Good. So our second presenter is local, running a project manager at the Biozentrum here in Basel. And Simon Ittig is also the founder of T3 Pharmaceuticals, correct? So you invented, let's say, worked on a new platform, Bacterial platform, to target or to bring the proteins directly to the cancer cells. Okay, you want to this one? Or? Okay.
Okay, so I feel that we're going to be a little the outgroup on this Nanomedicine conference. So the company is called T3 Pharma. It's a spin-off from the Biozentrum of the University Basel. And I'm looking forward to tell you something about our bacterial platform for delivery of active proteins into cancerous cells. So it's going to be about targeted cancer therapy, and then what are the characteristics of good targeted cancer therapy. And we said in the beginning, so it should allow the delivery of large amounts of proteins to the cancer cells while reducing the amount of proteins delivered to other sites, and at the best case should be versatile in types of proteins that such a system could deliver. And bacterial cancer therapy has been known for many, many, many years. So you see a not very recent issue of the New York Times here. So more than 100 years ago, this guy, Dr. Coley, he used live bacteria to treat cancer patients. But at that time, microbial biotechnology was very limited. And so they just had to use the bacteria that nature would supply them with. And I think that's very different nowadays. So at least to the same extent as photography had made progress, also genetic engineering has made. And so what we're using is genetically modified Yersinia enterocolytica. And with the genetic modification, we reduce the bacterial virulence. And at the same time, we aim at rendering these bacteria more specific for tumor cells. So the question is how, by genetic engineering, to target the bacteria to a solid tumor. And this is built on a negative selection. So it's known that the solid tumor represents a site where the immune system is very weakly active. And by depleting the bacteria's intrinsic ability to fight the immune system, you render them like specific for the cancer via the negative selection. So the immune system will kill the bacteria wherever the immune system is active. At the solid tumor site, the immune system is predicted to be weakly active, so it cannot get rid of the bacteria. That was our initial hypothesis, which we then went on testing in mouse models of cancer. So we used syngeneic models where the immune system is normally active in the mice. We did this in collaboration with Gerhard Christofori, who just had a talk in the morning before. We have a tumor in the flank of the mouse growing. We administer live bacteria intravenously to the tail vein, and then over several days we follow where do we find how much live bacterial counts in the organs. So we do really organ sampling, dilution and plating, and count live bacteria per gram of organ. You see here an example of a melanoma mouse model for a wild type strain, and then our engineered strain. And what we count is the live bacteria per gram of tumor or per milliliter of blood. And you see that the wild type strain, as used like 100 years before, it has a very good distribution in the tissue. So you see high counts in the spleen, which is known for Yersinia to occur, and then diverse counts in liver, lung, and tumor, and the same at day 2. And you see that upon genetic engineering, we manage to have at day 1, upon intravenous administration, low counts in spleen, liver, lung, no detectable counts in the blood, and already starting to have somewhat close to 1 million live bacteria per gram of a solid tumor. And then you see at day 4, this gets even more pronounced. So the bacteria keep on growing in the solid tumor and reach more than 100 million live bacteria per gram of solid tumor, which is very high. So meaning that we have accumulation at the solid tumor site finally of more than 1 million-fold as compared to our detection limit for live bacteria. And I can tell you that this colonization keeps on being the same for up to 2 weeks. So the question is, why do we use this Yersinia? I hope that the data before could kind of argue in favor of using Yersinia over other bacteria that had been used before. So the targeting to the tumor is superior than what has been shown in literature before. And then there are additional features of this Yersinia which render them very good for application in patients, so for toxicity, for the spreading, and then the animal to human transition, which can be tricky with live bacteria. I think what is very interesting about this negative selection, bringing the bacteria to the solid tumor site, that it's not built on any knowledge on cancer epitopes which need to be present. So it's built on hallmarks of solid tumor physiology, that the vasculature is high and that the immune system is weakly active. So this, we have tested some cancer models, and we are planning to extend this to see that this can hold true for many different solid tumor types. So having said that we have these bacteria that can go and grow in a solid tumor, it's the question, how do we get them active to bring a therapeutic substance to the cancer cells? And there we have developed a technology which is built on a bacterial nanomachine that allows the delivery of therapeutic proteins from the bacteria right into eukaryotic cells. In this way, we introduce pro-apoptotic protein as well as other mode of action in the field of immuno-oncology. So some more words about this proprietary platform for protein delivery. I think it's very interesting, or would be very tempting to deliver active peptides or even proteins into eukaryotic cells, and as you all know, due to the membrane physiology, that can't be a little tricky. We use this bacterial system that you see schematically shown here. So the bacteria have this system by nature. It's really a thin needle at the periphery of the bacteria that is normally used by the bacteria to deliver bacterial proteins highly selectively into eukaryotic cells. And what we do is just we make use of this multi-protein complex system to deliver therapeutically interesting proteins, and not the proteins the bacteria were aiming at delivering. So this system is called type 3 secretion system, giving the name to our company. And then by this way, it's clear in the context of cancer that we are aiming at impacting the cancerous cells by delivering active proteins. So I think it's very interesting that the bacteria, they do not only deliver these proteins right into the cytosol of a cancer cell, but they also produce them themselves. So it's kind of a factory that goes to the tumor, replicates there, produces a therapeutic active protein of choice, and delivers it right away. We started applying this in cell culture, so we had the bacteria on top of adherent cells, and we delivered a peptide that could easily be stained by immunofluorescence, shown in green here, and then you see that within 1 hour after bacterial addition, you get quite high signal, it's very homogeneous, and you reach almost all of the cells. It holds true for many different cell lines. We so far have tested more than 140 different proteins for delivery by this bacterial nanosystem, and we published this end of last year. So I think an important question in this context is that how much protein can be delivered by such bacteria. We assayed this again in cell culture, and we could see that delivering eukaryotic proteins like cell cycle regulator INC4C or apoptosis-related protein BIT, you see that's the delivered protein, and these are the endogenous proteins from the cell. From proteomic study, you know that the endogenous band in this case corresponds to 500,000 protein copies per single cell. So using the bacteria, you can deliver more than a few million protein copies into a single cell in cell culture. So that's about the number you can get. So I'm going to show you one example of apoptosis induction using this bacterial protein delivery system. I think we started working with endogenous human or murine proteins that were known to induce apoptosis, and we further improved them by genetic engineering. And you will see on this little movie on the left side, we treated HeLa cells that express actin GFP for visualization with doxorubicin, the classical chemotherapeutic agent. On the right side, we added our bacteria, delivering one of our lead peptides. And then time is in hours and minutes. And you see that on this side, very rapidly, the protein goes into the cells and has a strong effect on cell viability, which holds true for many different cell lines. And I can also say that some peptides we know have activity on the cells within minutes. So that protein transport is really rapid. So I think our company has two platforms. One is the tumor targeting bacteria, which we then further equip with this protein delivery technology to bring therapeutic active proteins to cancer cells, and we're working on getting their efficacy studies in mice. And I think this allows us to get additional assets like these proprietary therapeutic proteins, which increase the efficacy on a given pathway. With this, I would like to thank our small team, Christoph Marlis and Ausma, our collaborators in Basel, Erich, Nick, Gerard Christofori and Guy Cornelis in Belgium, from the FMI, Mark Bühler, and then our funding, and last but not least, the University of Basel and the Biozentrum. And I thank you for being here today.
Thank you. Excellent talk. That will work also in metastasis?
Depending on the size, we see that we get some metastases that are colonized by the bacteria. I think it needs these hallmarks, so if it's a clump of very few cells, this might not hold true. But upon a certain size, this will work or has been shown to be colonized.
Hi, I was wondering if there are any non-pathogenic intracellular bacteria that you could use for this technology to kill the tumor cells from within.
I mean, those Yersinia, they stay extracellularly. There, in the clinical trials in the US, are intracellular bacteria that they use, but it's even more pathogenic than the Yersinia. There are people trying to equip E. coli. I mean, some pathogenic E. coli do have this type 3 secretion system. There are some people trying to bring it actively into E. coli that were not supposed to have it. I guess it can work, but it's always difficult. It was not meant to be, so it's not very easy to bring the system to other bugs. And I think if the bacteria haven't been made to persist in the bloodstream, they can be cleared very rapidly. So there need to be a certain degree of virulence, but clearly not too much.
Thank you. That was very interesting. Could you use something? Use it to deliver something else than proteins, like siRNA, microRNA?
Yeah, we're trying with Mark Bühler from the FMI to have RNA delivered. This system is really purely made for proteins, but we're still going to try it. There is a similar system that has been shown to deliver DNA, and we're also going to implement that.
Any other question? I have a question to you. I mean, it's maybe just an estimation. When would you say how long it takes that it really could be used, let's say, in the clinical setting?
I mean, as we have a company, we need probably to aim high. So I would say 2019 or so we're aiming at. So we're going really now full speed into efficacy studies in mice. We're trying to see the way for a regulatory perspective, what would be needed, and then we...
Okay, so but 2019 is not so far away.
No, I mean, it's good that there are some companies, even in Basel, Switzerland, that use live bacteria for cancer treatment, and then some in the US which use formerly pathogenic bacteria intravenously administered to cancer patients. So it's not that we would be the first one doing this. Well, the mode of action is different.
Okay. Yeah, you mentioned before that you do already animal testing. So when do you start clinical trials for humans?
As soon as we can get there.
Okay.
Well, it's hard to predict.
Okay, good. Thank you very much. Very nice and interesting lecture. Thank you.